So it's my pleasure to introduce Mike, who's the Chief Scientific Officer of PathAI. Mike joined PathAI from Bristol Myers Squibb, where he led the clinical biomarker laboratories for the translational medicine division. Mike was a co-founder of Omnics, which many of you are familiar with, and this was a digital pathology joint venture between GE Healthcare and Pittsburgh Medical Center. And he was also lead inventor of the multi-omics multiplexing technology at GE. Mike has been very involved with Digital Pathology Association, having been a past president and serving on their executive committee, and led initiatives to develop industry-wide regulatory guidance. And so I think we're all looking forward to hearing from Mike, who's going to be talking about AI-based pathology in clinical stage biopharmaceutical drug development.
Awesome. Thanks, Lee. Can you guys all hear me, I hope?
Yep.
Great. Hey, so it's great to be here. Thanks for inviting me. I think it was a slightly short notice, but hopefully I pulled together a talk that you guys will find interesting. Let's see if I can share this. Can you guys see this? Everybody seeing this okay?
Yeah, we can see it.
Let me.
Mike, you've got the wrong. There you go.
There we go. How's that work? Hey, how you doing, Mike? And Anil, some friendly faces, it sounds like, or voices I hear on the phone. So great. So I'm going to talk today a little bit about AI-based pathology in the biopharmaceutical setting, particularly in the clinical trial or translational medicine space. I don't have any disclosures other than the fact that I work now at PathAI. So we'll go through kind of a brief intro on AI and pathology in drug development. And then I'm going to try to get into pretty quickly some examples of how we use pathology, AI-based pathology in oncology, in the PD-L1 story. Hopefully I'll get through the Cd8 topology story really quickly, and then get into some non-oncology work that we've been doing in the liver NASH space. And then I have one slide at the end. Hopefully I'll get to that as well, just to, I'm sure folks here are aware of it, but just to kind of paint a little bit of a picture on the regulatory environment. So artificial intelligence-based image analysis has advantages over microscopy-based pathology, as I'm sure everybody here understands, that it can have higher accuracy in relation to manual microscopy, higher reproducibility, intra and inter-reader reproducibility, and because of that can be more heavily standardized. And I think importantly too, it's scalable. As we just heard, there's much more high dimensional data that we can get in the digital space than we can get in the manual microscopy space. And in the biopharmaceutical setting, this is particularly relevant when we want to make inferences about biomarkers and drug responses across large patient cohorts. So the opportunities for AI is, I can kind of break it down into both of these industry areas, biopharmaceutical as well as clinical diagnostics, but I think that the value drivers are very much the same across them, including, as I said, improving quality, accuracy, certainly improving workflow and turnaround time and costs are relevant in both settings, archiving. In the clinical diagnostic settings, there are, we have additional sort of value drivers in being able to do things like remote coverage and also do things like basic and translational research, which is in the biopharmaceutical setting really at their core. The utility drivers are slightly different. I think this is important in the biopharmaceutical space. Improving or increasing the probability of regulatory and technical success is largely the name of the game. And if one is able to do that, then you're naturally improving patient outcomes. Improving patient outcomes in a biopharmaceutical setting can translate obviously into clinic and improving patients' lives in a healthcare setting. And of course, there's others utility. And in the diagnostic space, actually, I should say, there is a drive in utility to improve patient outcomes from having better pathology, but also there is additional utility in sort of cost and lab quality drivers as well. I think in the just to sort of zoom out a little bit in the pharma space, AI and pathology can kind of play a role across the entire continuum from discovery all the way through to development. And so there's many areas of opportunity in which this technology can have advantages. I think in the translational medicine, particularly, there are unique opportunities where AI can play a role. And just kind of honing in on the translational space, we like to break it up into the exploratory space, which is this sort of retrospective analysis of clinical trial samples to look for things like novel targets or try to understand mechanisms of action better, certainly to generate plenty of hypotheses around patient selection and stratification, and using this information to help inform your trial design and your indication selection as you move into early trials. The technology also can have a pretty good role to play in the perspective, both early trials and late trials, doing a lot of the similar stuff that you would do in the exploratory phase, but doing it in a much more robust way and a prospectively defined analysis type of manner, and then ultimately getting into late registrational trials where you're largely now talking about selection and stratification. And there are multiple avenues to pursue for the technology in that space, including FDA clearances, lab developed tests under CLIA settings, or even the drug development tool, which is a biomarker qualification program at the FDA, is also relevant. I'll talk really briefly about that kind of toward the end. Pathology itself, even in the manual space, is traditional microscopy has a very large role to play in the biopharmaceutical setting, both in enrollment, selection, pharmacodynamics, stratification, of course, as we all know, and then these efficacy endpoints. And I'm just highlighting here toward the end, efficacy, using pathology as an efficacy endpoint is a pretty big deal, because you really sort of want to get it right. And I'll touch on that when we talk a little bit about fibrosis and NAFLD scoring in the NASH space. Sorry. One last thing I want to touch on that kind of sets up some of the data is that it's important to make the distinctions between clinical validation or analytical validation, clinical validation, as well as the clinical utility. In that when we're doing clinical validation, we're sort of testing whether, of course, the tests work well. Clinical utility is really if they're improving patient outcome in the end of the day. And a lot of our image analysis and medical imaging, whether it's radiology or pathology, has a tendency to stop at the clinical validation of the accuracy. How well does the test do against a manual reader? And that is our consideration of accuracy. So, and there's a reason for that. It's because utility studies are really time consuming, and getting well curated large data sets for outcomes are needed if you wanted to, particularly in randomized control trials, if you want to really look at the differences between manual microscopy and AI-based pathology, or AI-based ML-based imaging, to show that it has utility. So, and that presents a particular problem, because if you're really only training and validating against gold standard readers, and why would we expect superior performance? So what we've really been trying to do, both as an industry and certainly at PathAI now, is to try to demonstrate the utility in a setting of randomized controlled trials, to demonstrate that the AI has advantages over manual microscopy. So I'll show you some examples of that. Using PD-L1 is the first story to tell. So I'm sure many folks have heard now of PD-L1 and its expression as a biomarker associated with clinical responses to checkpoint inhibitors, to both anti-PD-1 and anti-PD-L1 checkpoints in multiple tumor indications, in lung, melanoma, head and neck, bladder, gastric, and triple negative breast recently. PD-L1 IHC assays have been approved as complementary or companion diagnostics. And the scoring algorithms with their clinically relevant cut points have been established for various tumor types and lines of therapy. I'm just showing here the PD-L1 approved tests that are approved for non-small cell lung, but there are, as I said, many other approved ones, and for multiple indications, first and second line now, et cetera. There are certainly challenges with the standard IHC PD-L1 testing. This was a paper that came out of Janice Taub's group, which is a really great story about how PD-L1 testing using manual microscopy is somewhat substandard in comparison to other types of biomarkers that are more quantitative, like tumor mutational burden or gene expression profiling using RNA technology, or even multiplex immunofluorescence, can have better areas under the curve as compared to PD-L1 IHC testing to predict patient responses. And there are many reasons why this could be the case for PD-L1. It could be sampling bias, both spatial sampling bias from taking biopsies, as well as the temporal bias in terms of when you're actually measuring PD-L1 versus when its activity is relevant in vivo. But there are multiple assays and multiple cut points, multiple clones and multiple reading scoring algorithms the significance of tumor cell versus immune cell is different. And so this all sort of lends itself to being somewhat of a substandard test. We look to sort of address just this issue of standardization of it, the sort of repeatability of it, and the accuracy in relation to actually being able to pinpoint the PD-L1 on tumor cells to see if that would have any impact in improving any of the efficacy scoring and any of the sensitivity as well. So we developed an AI-based model, and here in the bottom I'm showing the relationship between the prevalence and the efficacy, and we tested it in Nevo monotherapies versus manual microscopy. So this has now been published. We first published this at SITC a couple of years ago, but we developed a training model. We trained a model, I'm sorry, against PD-L1 stained slides. So we had a series of whole slide images. We got about 250,000 pathologist annotations to annotate tumor stroma and normal, and then to further annotate PD-L1 positivity in various cell types, particularly tumor cells, macrophages, and lymphocytes. So we were able to build a robust model that gave us PD-L1 cell types in these various compartments. We did a validation that we call a frames-based validation. We also published on this. Importantly, we did not validate against the whole slide image because, as I said earlier, we wanted to see whether or not we could be any better than the whole slide PD-L1 scoring. So we validated against frames where we take 150 by 150 pixel sized images, and we give them to 5 pathologists, and they score each individual cell, and then we compare that to the algorithm score and come up with a cut point at which we believe the model is pretty robust, and then we test it on held out data sets to infer PD-L1 scoring. This is a little bit more in depth than how we do that frames-based validation, so that we collect 10,000 distinct frames, and we bucket them by the number of cell types, both positive and negative, so we get equal distributions. And this is just showing the validation at a frames level on how well the AI model compares to the consensus reader, and how well individual pathologists do on the frames base as well. So there's a bit of a spread, as you would expect, when individual readers are compared against themselves, and the model is pretty tight when we compare against a consensus of readers, both looking at tumor cells and immune cells. So the model performs pretty well. Then we took that model, we showed it to randomized controlled clinical trials that the model has never seen before. Here's 3 cohorts where we looked at CheckMate 67, 238, and 275. I apologize here, this is a typo. So the 275 is a bladder study, not melanoma. And the first 2 are melanoma trials. And so we run the PD-L1 score and those sample sets, and we looked at 2 different cut points. So the first thing we did is we said, let's look at the original manual scoring of those PD-L1 scores from those original patients and compare it to our score now at a whole slide level. And we see pretty decent, but not great, I should say, correlations between PD-L1 scores and the manual scoring. We're looking at Kendall's coefficients of 0.6, 0.59, and 0.57. In every case, if you give it a bit of a squint test, you can see the digital scores kind of up scores a little bit compared to the manual reader. And I can show that a little bit more clearly here in the table if you just look at the additional samples that are identified by digital reading only. So these are the added samples into the positive patient population who otherwise would have been called negative by manual. And then the opposite of patients who may have been called positive by manual and negative by digital. And there's much fewer of those. So we wound up seeing sort of, if this was a 2x2, we'd see off diagonals where we seem to be up-calling PD-L1 using the model. And so the next logical question was, does it matter? And what happens to those patients? Are those patients responders and non-responders? Is this actually more accurate with respect to the patient outcome? And so this is looking at hazard ratios for all 3 of those trials at the cut points of 1% and 5%. And in the blue are patients who are considered to be positive by both methods, and in the dark green are negative by both methods. So you can see, for example, in CheckMate 275, the overall survival, strong association of the PD-L1 positive populations with overall survival. The middle bar here, the point estimate in orange, are patients who were negative who became positive. So you can see that they're trending more in most cases toward a positive association of overall survival or regression-free survival, kind of indicating that patient population is likely to contain lots of responders. And in fact, when we ran this, the whole trial as if this was the test, we saw really no changes in the significance of the trial outcomes. So that would kind of indicate to us that we're bringing in patients who are balanced in the same way that the overall population was. So if you think about this from a pharma perspective, you have a superior test here. You have a test who can improve the prevalence of the biomarker positive population, increasing the opportunity to give drug to patients who really could use it, and you're not jeopardizing your trial statistics at all. So we developed an accurate model there. PD-L1 has identified patients who are classified as negative by manual, and those additional patients seem to have additional response rates, as would double positive patients. So we think that a digital score could have utility here compared to manual. So that's the first example. I'm going to go through the Cd8 topology story fairly quickly. I only have a couple slides on that. The background on this is Cd8 killer T cells are relevant as it relates to immuno-oncology. I think it's sort of the underpinning of a lot of the success that we've had in IO lately over the past many years. And there's been, and this has actually been out for this idea of CD8 rich tumors and CD8 totally negative tumors, or so-called cold tumors, as being not responsive to IO therapies. That idea has been out for a long time, but there are additional phenotypes kind of in between. There are these so-called Brisk phenotypes where CD8 is associated with enhanced survival in response to IO, and then the non-Brisk, which we would consider like excluded or cold tumors. And so we were interested, this is back when I was at BMS as well, interested to understand whether this excluded phenotype have some sort of biological relevance as it relates to IO therapies. So we, again, that would be sort of difficult to do manually just because of the scale and the number of cohorts that we wanted to look at. So we built an AI-based model that can identify CD8 positive T cells. I'm going to take it one step further here quickly. And then we were able to take a patient cohort and set cut points in terms of having whether or not patients had a lot of CD8 that was primarily in the stroma or primarily in the parenchyma of the tumor, and could label them as inflamed or excluded. And then we would look at the gene expression profiles of each of those individual patient subgroups. And what we found is that CD8 patients that had predominantly CD8 in the stroma, which are more excluded and not predominantly in the parenchyma, have gene association patterns that associate largely with upregulated CSF1R. And in the opposite case, where we have a lot of parenchymal CD8, we have CDSF1R as being heavily downregulated. CSF1R is a gene that's been associated with macrophages and stromal components of resistance. And so it started to kind of make a little bit of sense to us that this excluded phenotype is relevant. Interestingly, the second gene that's upregulated in both cases is CD8. So you do have a lot of CD8 in both cases. If we were only looking at CD8 and not really looking at where CD8 was located, we might not see these changes in some of the mechanisms that we were hypothesizing could be relevant. So natural extension of that work is then to look into some of the cohorts retrospectively and see if this had anything to do with true resistance against some of the IO therapies. And that work I know is ongoing. So last part of the talk here, I'm going to switch gears a little bit and talk about liver disease and non-alcoholic steatohepatitis. So there's a growing sort of epidemic of non-alcoholic fatty liver disease. Prevalence is fairly large across the globe and is correlated with incidences of obesity and correlated with other metabolic diseases as well. There's an increasing and a projected increasing burden of this disease that's supposed to double over the next decade, particularly in patients that have severe fibrosis and moving into cirrhosis as well, and who would be requiring liver transplants. So NAFLD or non-alcoholic fatty liver diseases some population of that group can progress into non-alcoholic steatohepatitis with accumulation of fat and other histological measures in the liver, and that ultimately can result in cirrhosis. There's not a lot of therapy for NASH patients. This is a very large unmet clinical need, is to find therapies that can either halt the progression to NASH and or reverse the effects of NASH. NASH is one of these diseases that is generally defined histologically. I've likened this in my prior world to Alzheimer's disease, which is the definitive diagnosis is made histologically, and that is the case with NASH. So because the the histology is so defining for the disease, and also because the disease progression can take such a long period of time to result in clinical poor clinical outcomes. The FDA has issued guidances in terms of accelerated approvals for drugs that would be based on the histology or histological endpoints. So this is super relevant. So what the FDA has laid out here, again, because of the slow progression of NASH, liver histological improvements as endpoints is reasonable to predict clinical benefit. So here they've come out and said this can be a surrogate endpoint. The way they define these endpoints is that the resolution of steatohepatitis, there's two ways that you can have a drug be successful here. You can either have resolution of steatohepatitis with no worsening of fibrosis, or you can have improvements in fibrosis with no worsening of the steatohepatitis, or you can have both. So they look at these two key histological aspects of the disease and say, if you stop one and reverse the other, you'd be in good shape for an accelerated drug approval. The challenge is measuring histologically by manual microscopy the components of NASH, which is steatosis, lobular inflammation, and ballooning, is highly not reproducible. So these are kappa statistics looking at 100 and some odd, 150 plus biopsies across several pathologists, looking at their kappa, their agreements essentially at how well they agree with steatosis, lobular inflammation, and ballooning, and they don't agree very well at all. So the high range of reader variability can contribute to some of the challenges that we've seen in some of these NASH trials. This, and just to sort of put a pin in that, there was a paper that looked at theoretically what would trial endpoints look like if we had much less reader intervariability versus what we have now. So they ran 10,000 simulated trials with scenarios of having a true response rate of 20%, where the active compound was giving a 20% response rate and a 10% response rate with the placebo. And when you have perfect agreement in these components of NASH, you'd have the probability of finding drug effect would be 93%. But when you have the realistic interreader variability laced into these trials, you have a probability of finding the real drug effect to be 61%. So this isn't, I mean, anybody who's familiar with like Six Sigma or Gauge R&Rs, this is what you would expect. If you have a poor measurement tool, you're not going to you can only measure real differences so much. So interestingly, this does seem to be bearing out in these drug trials. So there have been some spectacular failures over the past couple of years with Intercept and GenFit and Sirius, where a study hit one out of the two recommended histological endpoints. The expectation that it would be enough for approval, but it wasn't, because the agency, basically did not agree, and they had some concerns over the variability of the histology there. Here's another quote: although the response rates observed with this treatment were broadly aligned with our expectations, the placebo response rate was significant. So large response rates in placebo are a problem, probably because of this interreader variability issue. And here's the interreader unweighted kappas of 0.4, 0.3 in NAS resolution without worsening, and 0.3 for fibrosis. So poor reliability of the biopsy allowed for improper entry into the study and misclassification for the treatment effect. So these are really big problems. So PathAI, this is before I got there too, developed models, AI-based models, to see if this interreader could really improve things. I'm going to accelerate here my speaking. So we developed two kinds of models. One is a human interpretable feature-based model where we do annotations. We have hundreds of pathologists who do this for us. And then we developed a model where we can infer ballooning, steatosis, and inflammation. And then for fibrosis, we did an end-to-end sort of a black box model where we just fed the model fibrosis scores and had the model come up with the model's fibrosis score. So this is just showing a quick validation of how well this compares to manual reading, pretty well for the NAFLD components. This is ballooning, steatosis, and inflammation. For fibrosis, this is again showing against manual readers how well we compare, how the model compares pretty well. Interestingly, we can also get kind of a distribution of fibrosis. Instead of getting a single fibrosis score, we can get a distribution of fibrosis across the biopsy here. This is just some old work too, just showing that the machine learning based scores can predict clinical outcomes, both in patients who are F3 or F4 with cirrhosis, predicting clinical events, and all of the components of the scores from the machine learning are able at a median cut point to predict clinical events. So let me just show you really quickly the trial some of the trial data that came from the top line results from the Atlas trial. This is Gilead's phase 2 trial, looking at multiple drug combinations, Silofur, and then combinations of those. And they're looking to see whether there's a one-stage improvement in fibrosis or progression to cirrhosis. And the best drug cocktail is the Silofur arm, which showed that there were 21% of patients who had a one-stage improvement in NASH, in fibrosis, I'm sorry. But again, pretty decent placebo group here had a improvement in NASH, and that was not statistically significant for this particular trial. When we went and looked at the machine learning fibrosis score, looking at the same arm with the combo Silofur, we see a significant improvement in the resolution of NASH compared to placebo. We also see a significant change in the reduction of F4 fibrosis and a significant improvement in the F2 fibrosis for those patients in that arm where they suspected to have the best drug effect. So here we see significance using the manual methods. They don't. And now kind of a logical question on this is a little bit different because we're looking at linear scores or quantitative scores. What if we extrapolate what a ordinal score would be in the same way a pathologist does it? And then if we redo the trial statistics, do we see significance there? So that's work that is ongoing that we hope to be able to present soon. But nonetheless, we think the machine learning tools here are the way to go. And that's the last thing I'll say. I think on the regulatory space, there's been huge, great improvements from the FDA, both for primary diagnosis and just clearances in general, and also giving draft guidances. Most recently, they've proposed a framework for AI and ML tools for software as a medical device, and the FDA biomarker qualification program is starting to get very interested in machine learning to be using these technologies as endpoints. And here I'm just listing 3 separate guidances from the FDA. I already talked about NASH, but the same principle applies for complete pathological response in neoadjuvant setting, as well as mucosal healing in the ulcerative colitis setting, all requiring histological endpoints that would be relevant for drug approvals. So that was a lot. I know I only gave 2 minutes here, but I hope I was able to sort of impress upon the team here just how using machine-based learning can have really great improvements in the utility and improving the regulatory and technical success for drug programs. So I'll stop there.
Mike, what a whirlwind.
Yeah, a lot of stuff there, Mike.
Glad to have you back home in the
Thanks, I appreciate it. Glad to be back. So I know we're short on time, and I'm happy to take questions through the channel here or through email. I know it's a lot of material, and I can certainly share a lot of the posters, presentations too, if people want to dig in more deeply.
So Mike, since nobody's posting you a question, how can we as vendors in the community help liberate the data and broadcast to the pathology community the needs we have in a more effective fashion with the new seat and new hat you're wearing?
How can we say that one more time? Like how can we liberate the data for the pathology community?
So how can the startup company community better engage the pathology community at this stage of development with the growing excitement for the use of these tools in pathology practice and in
It's hard. I mean, how do we better do that? I think, that's a super good question. There's a lot of evangelizing, I think, that we've all been doing, both for the pathology community, but I also think in the pharma community. I mean, I've sort of targeted a lot in the pharma community, particularly in this translational space. It's funny, Mike, a lot of the challenges that we see in the pathology community in terms of having to create believers also exists in the pharma community. And the reason I think that's relevant is because a lot of great technology in the biomarker space and comes out of pharma oftentimes. TMB is like a really good example, and there are others. And I think if this gets a better foothold in the clinical translational space in pharma, I think that you'll also start to see some things sort of lightening up in terms of getting the pathology community to sort of realize the value of this to some extent. So, but conferences like this, I think, are helpful.